just as out of curiosity, Sarah and I did our fellowship under Dan Kastner and Rafaela Golbach-Mansky and Ivona Aksentievich, whom many of you probably know well. And this was in the early 2000s when NOMID, which didn't even have a name at that time, was endemic for the NIH. And we're certainly excited to be at this point and to see how much the field has advanced, the diseases that have been recognized, and also to see the pediatric rheumatology community together around these and the involvement of patients and parents for these diseases. This is really a huge step, at least from our perspective. Now, I was asked to give a talk on two aspects for the development. This is a standard disclaimer slide. There's nothing unique about it that we're required to present. What I would like to cover, hopefully quickly, is first the FDA programs that are designed to facilitate drug development for rare diseases, and then also talk about the clinical trial designs, considerations for rare diseases. And just that topic on its own may take a whole conference to talk about the nuts and bolts of clinical trial design, but I'll try to summarize it for you. The first slide is really to provide you some of the history of FDA and to get an idea about the framework under we function. And even though FDA was established in 1906, I think, the first landmark legislation that authorized FDA was in 1938, which is the Food, Drug, and Cosmetics Act, which authorized the FDA to require safety data for products to be approved or to be marketed. And another big landmark in the FDA history is the 1962 Kefauver Amendment, which followed the disaster, the thalidomide disaster, which authorized FDA to require also demonstration of efficacy, and not just efficacy, but substantial evidence of efficacy, which FDA has interpreted to mean adequate and well-controlled investigations. As you can see, S is underscored because this usually, the FDA has interpreted this to mean at least 2 well-designed and adequate clinical trials. So I'll touch a bit on that in the context of rare diseases because we may have some regulatory flexibility of how we interpret that. And also the Congress has recognized the need to facilitate drug development for rare diseases, and it passed in 1983 the Orphan Drug Act. And this was partly to address some of the challenges which we discussed today, and some of these are listed on this slide, are from the small populations. In some cases, ultra-rare diseases, really, really small populations. We're talking about hundreds at the most patients from the world. In addition, as we all know, many of these diseases are heterogeneous, and even though the underlying pathophysiology might be the same, the clinical manifestations might be completely different. So capturing this in a clinical trial might be really challenging. Also, the natural history for many of these diseases is not well understood, particularly the more recently recognized. We don't know really, I don't want to go into details, but these are some of the considerations for the clinical trial designs for rare diseases. Also, for most of these, we don't have regulatory precedent. There are a few exceptions this far. And I think Christina touched on the selection of endpoints, which may be very, very challenging for rare diseases. So with the passing of the Orphan Drug Act in 1983, the FDA established the Office of Orphan Product Development, which was again designed or established to facilitate drug development for rare diseases. And this is the office that grants the designation of orphan drugs. Now, just to point out that this designation is not automatic. In other words, the sponsors have to apply for orphan drug designation. And once designation is granted for a specific product and for a specific indication, this has certain benefits for the sponsor, which can stimulate further drug development in that area. And one of them is that these sponsors or these applications for these products are not subject to user fee when the application is submitted, which is not a negligible amount. In addition, there is a post-approval marketing exclusivity for 7 years, and also no pediatric studies under PRIA, which are required for regular products, are required for the drugs developed for rare diseases. So again, this is intended to minimize the upfront cost for developers to stimulate investigations in rare diseases. The Office of Orphan Products also provides grants and funding for studying natural history of these rare diseases, as well as grants for studying safety and efficacy of these products. And just as an additional perk to sponsors who develop products for rare diseases, there's this so-called Rare Pediatric Disease Priority Review Voucher, which means, and this was authorized by the FDASIA legislation, for sponsors who receive approval of drug for rare pediatric disease, they may qualify for a voucher for a priority review for another application, which might be for a much more common condition. Again, this is designed to expedite drug development for rare diseases. And there are some links that I provided there, might be helpful and useful for these programs that FDA has generated. Now, to further the development of not only rare diseases, but diseases for treatment of serious conditions, the agency has several other programs, and these are listed here, and I'll just summarize them on the next few slides quickly. As I mentioned, all of this should be for treatment of rare diseases, and the fast track designation is sort of a fairly low bar where the justification might be based on mechanistic or theoretical data or some non-clinical data in animal models of the disease or some clinical data. Now, the breakthrough designation, breakthrough therapy designation is certainly a higher bar for sponsors to meet, to be granted this designation, but that commits the agency to a lot more intensive guidance on how to develop these products. And to be granted a breakthrough designation, usually sponsors need to provide clinical evidence, can be preliminary clinical evidence, that the drug may demonstrate substantial improvement over existing therapies. Now, the fast track designation and the breakthrough designation are usually given during the early stages of development, again, to facilitate the interactions with the agency and our feedback or input on the development program. The next one is the accelerated approval, which is a program, again, for treatment of serious condition, where there might be a meaningful advantage over available therapies. And in this accelerated approval program, we rely on a surrogate marker to approve a product to make it available on the market, and a surrogate marker or intermediate clinical endpoint. And I will spend a little bit more time on this to try to make a distinction between a surrogate marker and what the standard clinical endpoint might be. For standard clinical development, we would expect that the efficacy is demonstrated on how a patient feels, functions, or survives. In some conditions where, again, we may need to get the drug earlier on the market, we may need to rely on surrogate markers. This is a very common approach in oncology, where the initial approval and accelerated approval relies on shrinking, for example, the tumor size, which patient doesn't feel. We don't know what it does to the patient. So, but we believe that there is a reason that the drug is doing something good to the patient. So this would be a surrogate marker. Usually these approvals are followed by confirmatory studies where we actually look at the hard endpoints, either survival, function, or clinical benefit. And the fourth program that the agency has developed to expedite or to incentivize development for these serious conditions is the priority review, which many of the rare diseases that we discussed today would fall under. And this is essentially condensing the review time from the standard 10 to 12-month clock to 6 months. So for some sponsors, it might be a big incentive to start marketing the product a few months earlier, and we make the product available to patients earlier. So during these interactions, the FDA is certainly open to provide feedback to developers of products for rare diseases. We have a pretty good experience with different trial designs, and we're certainly open to provide this guidance essentially to try to minimize the waste of patients and resources, given the small pool of patients available for these trials. So I think we certainly encourage anyone to use this expertise that we have. I mentioned that our standard for approval is that we need to have evidence of safety and effectiveness, and the effectiveness usually is interpreted as evidence from adequate and well-controlled investigations, not one. So I think for rare diseases where 2 adequate, well-controlled studies might not be feasible, we can certainly consider what else might be necessary to support an approval of a product. And we would still expect to have some evidence that the drug actually works, so that should come from a clinical study or some clinical data. Ideally, that should be a multi-center study, well-controlled with robust or persuasive statistical data. We also need to better understand what the biological plausibility or the pharmacokinetics and mechanism of action of the product is. And also we can rely on some biomarkers or pharmacodynamic endpoints to support a clinical efficacy seen in a single clinical study. We also may use the knowledge that we have for similar diseases or populations. I think the patients are in the unique position to provide invaluable input to both the drug developers and the regulatory agencies like the FDA on to identify areas of unmet need and also information that may help us identify the risk benefits in the disease that is being studied. So FDA has been committed to including the patient's perspective in drug development, and this has been embraced by the agencies as a whole. There are formal patient-focused drug development groups on different diseases that have been even mandated by the so-called PDUFA negotiations, which is beyond the point of today's talk. But I just wanted to emphasize that patient input is critical for the drug development. And there are several examples, successful examples of patient and parent advocacy groups that have significant impact on drug development, including the funding, clinical outcome assessment, patient recruitment, and so on. So we're certainly excited to see that patients are involved indeed in today's meeting. So with this, I would like to switch gears a little bit. And this is just a reminder of the challenges and the considerations for the clinical trial development or clinical trial design for rare diseases. And specifically, the challenge is how to derive adequate evidence of efficacy and safety with the limited number of patients that we have. The rare diseases can be completely different. Endpoints might be completely different. Drugs might be completely different. So there is no one single study design that would fit all of these. And usually this may require a different design for different indications. So I'll present on the next few slides. Yeah. Well, I realize that I have only 1 minute, but I'm the last speaker, right? Yeah. And the dinner is at 6:30? Yeah, and the dinner is at 6:30, right? Yeah. Oh, we have time until 7 then. So there are multiple considerations when we talk about rare diseases, and including the endpoints, and also consider whether the disease should be treated chronically or intermittently, and what kind of dosing should be used. So these are all questions that we need to ask ourselves. Certainly when we review these, we need to have answers for these to inform prescribers if we are to approve a drug. So starting with the most traditional randomized placebo-controlled study, which is a pretty straightforward design. This is might not be applicable for many of the really rare diseases, but we have good examples where this was used. For example, the core trial that searched for approval of tocilizumab was a placebo-controlled, randomized placebo-controlled trial. I just mentioned the pros and cons of this of each of these study designs. Again, this is probably the cleanest and gold standard for assessment of efficacy. This is a trial where we can estimate at least a real treatment effect size. There are certainly cons. One of these might be that it's unethical to keep patients on placebo for a prolonged period of time, which may need to have provisions for early escape. And in this situation, this may compromise the interpretability of the results of the studies. If the escape is early and and significant number of patients escape. So the second study design that I wanted to share with you is the randomized withdrawal design, which has been popular with rare diseases, pediatric diseases, particularly polyarticular JA studies. Many of these have been designed as randomized withdrawal studies where you have an open-label lead-in period, and after that, the responders only are randomized to either continue on the drug or stop the drug in a blinded fashion. And this is sort of the readouts, and we're assessing the return of symptoms or flares. And this is some of the readout that can be used for such trials. Again, these trials have its pluses and minuses, and I don't want to go into much detail about this. But at least it minimizes the exposure to placebo because patients get retreated if they flare. The problem with the study is that it's hard to assess safety because all of the patients were exposed to the drug to begin with, and we don't really have a good sense of treatment effect size for these trials. So these are some of the limitations. Now, for a disease that is highly symptomatic and you can assess symptomatic response quickly, and for a drug with short half-life, a crossover study design might not be unreasonable. And we also have good examples of where this was used. I think a similar design was used for the approval of anakinra for NOMID, where patients served as their own control, and that minimizes the variability within the intra-individual variability. So again, this has limited application because you need short-acting drug like anakinra that can be washed out, and also symptomatic disease that would go away with treatment. So a placebo phase design, which we haven't seen a whole lot, but it's also another possibility to assess potential efficacy, is where you phase in treatment. And this is the type of response that you would expect. Again, this is I summarized the pros and cons, some of the pros and cons of such study design. Essentially, all the patients would get treated at some point. And a study design that I wanted to share with you for consideration is the dose-response design. I think Christina touched on this, which is not really on the radar of academic investigators and patients and parents, is that in addition to the safety and effectiveness, it's important for us to know what is the right dose to have an acceptable safety profile and efficacy. So one way to address this is to study different doses. Ideally, it should be a placebo-controlled dose range if it's ethical to administer placebo, and then you assess for exposure response. Again, I mentioned some of the pluses and minuses of this, but this is really important for us to inform what kind of dosing should be, not just nominal dosing, but also what kind of frequency should be used for these rare diseases. And that actually applies in general, not just for rare diseases. One non-inferiority design, which is also a possibility, implies that we have a pretty good estimate of the effect size of the comparator product, which is usually an approved product, and then we compare the effect of the investigational drug to that product. So we really need to have a good understanding of the effect size of the comparator to estimate the non-inferiority margin. Again, these might be too much of a detail, but again, these are considerations that are still practical. One issue might be that it might result in a slightly larger trial, but that would be a good way to compare it to both safety and efficacy to a benchmark. And I think I'm done with the sort of nuts and bolts of the trial designs. I'll be happy to entertain questions. But in summary, I think I summarized what kind of programs FDA has to incentivize drug development for rare diseases and also emphasize the importance of patient participation and parents' representative participation. And some of the study designs that we have successfully used and we are certainly willing to consider. And I think that's, yeah.